metabolite. You have 3 different reactions that can produce the metabolite. You have 2 different reactions that can consume. Pseudo steady state means whatever is coming in should go out. So, if you are designing a transport or an exchange, so basically let us say this is the cell, this is your like adjacent space near cell wall, and this is extracellular environment. When you are defining your transport, let us say we are doing it for this metabolite, and you will define the same metabolite with a subscript, let us say we call it AT for instance, and then you will define your exchange reaction without defining anything here. So, in that case, for A, obviously there will be at least one reaction that can produce A. So, you have one reaction that can produce A, you have one reaction that can consume A. So, you theoretically can get a flux as long as you have a well connected pathway. Same thing for AT, because you have one reaction to produce AT, you have one reaction to consume AT, that is it, your problem is solved. All these issues, as I said, for photon, for modeling the transport or exchange mechanism, sometimes you need to do that for the sake of modeling, and sometimes it does not really correlate what we actually observe in real scenario, but you need to do it because we operate on the pseudo steady state domain. Are we clear? Okay. One other question that I got is how to model an unknown microbe. You have an unknown microbe, how to model it. So, what is the first step? You have isolated a strain, for instance. As I said, when I was in Himadri's lab, one strain was isolated I think from a stream in the campus of UT Austin. Who would have thought that we could get a strain from that place? But someone fancied a thought, isolated the strain. What is the next step? Let us say for that strain, I would like to build a model. What is step number 1 or 0? Sequence alignment. If you could sequence the genome, at least you have some ideas like what are these genes, although they are not annotated. So, you have sequenced the genome. What is the next step? Annotation, but again it is an uncharacterized organism. Technically, yes, you can try to characterize each and every gene in your lab, but it will take years. For what are the tools we have so that we can annotate it? Might be of like medium or lower quality, but you can still do it. How can we do that? BLAST. Like you use the information from related organism and then you characterize the genes. From that genes you will get some functionalities. What will happen, as I mentioned, even for E. coli there are 35-40% genes with unknown or hypothetical functions. Now, for this kind of system this number or percentage would be way more, but you from that point onwards you can follow the similar steps and build the model. The issue that you will face, that you will not be able to use the tool that we discussed yesterday, K-base, most likely that is not there, but there is a way you can import this information and yet you can do that.
In the K-base.
Yeah. So, I do not know who wrote this. So, this is my oversight. I should have asked you to write your name so that I know who wrote what, but anyway you wrote it. So, does that answer your question? Okay. So, for today whatever you will write, please write your name, because I want to know who wrote what. Okay. So, these are the issues that I could see from yesterday's class. From the first class I got few other confusions, and I would like to talk about it before I move on. So, one of the questions that I got is how the products are impacting our life. Okay. I'm really glad that some of you appreciated my slides, but this is one of the issues. So, okay, we have talked about 4 different examples, 4 different microbes. If you remember in the first, is it the first or second lecture? But the key question is, so what? That's the key question, right? So, well, we can use this microbe, we can design, we can grow, we can produce a useful product, but at the end of the day, how is that affecting or impacting our life? Unfortunately, for most of these studies, we are not there yet. Like there might be some studies in the pilot plant level, but when we are thinking about industrial level scaling up, you will not find many different applications. I think I mentioned about one example by Genomatica, butanediol. That's the best example I could come up with on industrial biotechnology in that scale. But I think this will change. I mean, you know, because we need to just understand the potential of this bug, not only this bug, the entire biotechnology field, and then we have to convince people, because this is something, at least for photosynthetic microbe, we are using some resources which are aplenty. So why not make use of them? So again, to answer your question, for many of these works, this kind of scale up didn't or hasn't happened yet, but I'm sure that that will happen down the line.
May I add?
Sure, please.
Okay. So this one thing I could suggest in terms of the bioproduction. So 95% of the cancer drugs comes from the plant. Okay. And if you go to a big company like an Novartis, so you will see thousands of liquid robots. What they're doing, so they're bringing lot of different plant material from basically Amazon rainforest. Okay, and they're actually extracting and putting on different. They have a different cancer cell assays and trying to find out, mining that which plant has some chemicals. Now let's say if there is a drug target, you can even if find. The question is getting that amount of the drug for all the cancer people. It's almost impossible because you have to clear the Amazon rainforest for that, or you cannot make those things in USA or India because there's a. So whole idea, if you can really think about, if you know that metabolic pathway, or you know the structure, if you can find out, if you really can do in the microbes, then you can do it. So it is not today's, but if you can think about, if you really targeting the cancer chemotherapeutic, so this will actually can decrease the cost like several orders of the magnitude. Even what he is getting or he is getting in the lab scale, which may not be good for succinic acid.
Yeah.
But that amount, if you get in
specialty chemical,
specialty chemicals, that will actually nail down things.
Yeah. Thanks. Actually, yeah, that's a very good point. I got one question about carbon and nitrogen fixation and how they are linked. Can anyone take that? I think by now you should have a better idea how these two are linked. How carbon and nitrogen fixation are linked? Not for all, because not all the cyanos can fix nitrogen, but let's say the ones that can fix nitrogen, how these processes, these two processes are limited or correlated actually. So you are saying we fix carbon, and when we fix carbon, we produce oxygen.
So, like the cyanobacteria is using carbon.
Yeah.
As the carbon source, and in between that, it is fixing nitrogen.
Yes, but not at the same time or not at the same compartment. I think the question is, we know some cyanobacteria could do that, but what's the actual relationship? So carbon dioxide fixation is an energy intensive process, but we need to, like those bugs need to do that, because then it could use the breakdown product for anabolism to produce the other useful things for its growth, but at the same time I think the answer lies mostly about how do you see carbon fixation and nitrogen fixation, like through what glasses, because both of them are energy intensive process, right? We know that. We know that to fix a carbon dioxide, let's say let's limit ourselves to cyanobacteria. So to fix carbon dioxide, we know that through Calvin-Mason cycle we need to have 9 ATP, 5 NADPH. To fix nitrogen, we have to spend 16 ATP. Both of them are energy intensive. So as such, carbon fixation and nitrogen fixation, they're limited, they are correlated in that way, because Carbon fixation occurs mostly in the light time for a unicellular organism like Cyanothyce. So, they produces carbon, they fixes carbon dioxide, they produce ATP NADPH so that they can power. Okay, let me, I think I have to draw something here. Okay, hold on. Okay, so here is the deal. So, CO2, and this is a very crude simplified diagram. We know these bugs can fix CO2, right? So, they will spend energy and CO2 will be stored in what? Let us say in glycogen. So, while storing glycogen, this glycogen will be used in the dark to power the metabolism, right. So, you could see that carbon dioxide is stored, and that stored carbon dioxide is basically used to fix nitrogen. Now, can you see the link? For the case of filamentous bacteria, which has a vegetative and a heterocyst, this thing occurs at the same time, but in different compartments. But the basic principle is the same. Does that answer the question? I don't know who wrote it, but who wrote it actually? But does that answer the question? What do you think? Is it clear how carbon and nitrogen fixation are related? Any question on that? Okay, I think one other question was, is there a detailed electron flow chart available for microbes? I think we have discussed about that. Are we clear on like different electron transport mechanism? And it's very clear in literature. First of all, you need to figure out what kind of microbes you are dealing with. If you can figure out what kind of, then I think you can definitely have a generic idea. Some of them have some exceptions, but excluding those, you can definitely have some references to begin with. All right. Okay, I got few other questions. So, I think, yeah, I'm glad that this question, someone raised this question. What are the maths that we use for the modeling or analysis? I think we have briefly discussed about it, right? What we have discussed, like when we are doing the pseudo steady state based analysis, we started with what? An ordinary differential equation. We set up the equation and then we said it's under pseudo steady state, the left hand side is 0, so it becomes a system of linear equations, plain and simple. We know that it's an underdetermined system, we have a degree of freedom. Based on that, we will fix or we will determine 2 or 3 variables depending on if the degree of freedom is 2 or 3, and then the rest of the variables we can just solve from the system of linear equations. That is it. Again, this is only for FBA. When you are dealing with a kinetic model or a complex model, then your mathematical framework will obviously be different. But for the sake of time and scope of this class, I limit myself to FBA so that I can give you at least some generic idea. So, that is the math involved. Are we clear on that? How could we identify new strain? I think the problem or the issue is stemmed to what's the need, right? So, we will do this exercise in today's class is what's the problem that you would like to solve? As I said, always the backward design, right? You need to solve a problem. For that, you need to find the best system. I think the question was, let's say, I think it's based on the third example that I showed, the Staphylococcus 7942 strain, which cannot produce isobutylaldehyde, if you remember. We need to knock in the gene. The question was, well, if the inherent gene cannot produce something, why bother engineering it? Can anyone take that question? Yeah. Oh, your question, okay. Can anyone answer that question? So, the question was, if a native strain is not capable of doing something that we are interested in, why bother doing it? Why not, like, because we borrowed the gene from a different organism, right? Why not we use that platform to produce that product? So, you are saying about growth rate, and you?
Maybe it is not possible to culture that organism
issue with the culture. Okay, what else?
Maybe one of the reactions limiting rather fast.
Yeah, one of the reactions is limiting. All these are valid, but above everything,
Maybe there is some byproducts of that also.
there could be byproducts. So we are talking about many different issues. Yeah.
Like that product that we are interested in, it is in some other microorganism.
Mm-hmm.
We can put the gene in the like photosynthetic microorganism, so you can utilize the light energy instead of.
Eh, eh.
But so we can use another photosynthetic organism also.
Hmm.
So it is possible.
Yeah. So I think we addressed all the issues here, but the issue with their system was they would like to use carbon dioxide and light, which is why they picked the organism. Now, at that point of time, the reason for picking that organism was that was the fastest growing cyanobacteria. And again, you might laugh that fastest growing, like the doubling time is 2.5 hours compared to E. coli, pretty slow. But that was the fastest growing strain at that point of time, for which we knew the synthetic biology tools we could have engineered easily. So whenever you are so basically you are solving a design problem, right? Picking a platform, that's always tricky, but that also have some constraints. So if I can pick now, I mentioned about the fast growing strain, which has a doubling time of 1.5 hour, but that strain is still being characterized. So hypothetically, when the strain will be characterized, we will have more information on the strain. We will have more tools developed for the strain, and then using that strain as a platform will be easier. So many times that determines like what kind of system you are picking. So not only what is the source it's coming from, but what is your objective. If your objective is to utilize renewable resources like carbon dioxide or light, then you need to pick a photosynthetic organism. If you are picking a photosynthetic organism, you better pick an organism which grows fast. You pick an organism for which you have more knowledge. You pick an organism for which you have more tools. These are logical steps, right? Does that answer your question? Okay. Okay. How do we define efficiency as a selection criteria? So I said, I might have mentioned is efficient strain. What's efficiency? How do you define efficiency? I think I'm standing too much there. Yeah, I'll walk. What is efficiency? Like, how do you define efficient strain?
Desired product, high yield of energy, can produce a product quickly.
Rate, yield, how much, by consuming how much, right? So yeah, exactly. So when you are picking an efficient strain, as I said, yield, growth rate, these are the things you need to consider to characterize if a strain is efficient or not. That's typically how it's done. Any question on that? One other question that I got is, how do you define the rate constant? So for the sake of flux balance analysis, as if you remember, we completely got rid of this rate constant business. We are not doing that business anymore if we are doing flux balance analysis, because the concept is flow through a metabolite, which we define as flux. So we will just determine the flux, which will be a representation of the rate, but not the rate equation that we know of. But if you are developing a kinetic model, then obviously you need to know the rate. And I think some of you have asked me the question, and I sent you a paper how to determine that. So again, I have to define the class this way so that I can stick to the schedule. That itself is like a 1 or 2 hour lecture, and I wish I could do that. Okay. The other question was, we discussed about the push and pull mechanism, right? When we discussed the some of the lipid biomolecules that was produced in Yerobia lipolytica. So the question was, how many such changes we can make? Can anyone guess how many such changes and where to make such changes? How can we decide on that? I think I briefly discussed about it, but can you guess? How can we define which gene to push or which gene to upregulate or which gene to pull or which like how can we decide, or what's the limit actually? Can we upregulate like 5 genes for push, 5 genes for pull? What about 10? What about 20? Can anyone guess? Can anyone share the experience if you have already done it in the lab? So, yeah, I heard a whisper. Is someone thinking about it?
Like we need to choose the rate limiting step of the
Right.
so choose the push and pull the rate limiting steps.
Right. So, if you can determine the rate limiting step by any means, you pick those. Yeah, this is possibly the simplest thing you could do. Sometimes we have a way of doing it, like we have a way of knowing it, like which is the rate limiting step, right? So, you might have done some early experiments, and based on that you have some ideas probably this is the rate limiting step, or you can figure that by doing some modeling. A model will tell you the most likely these are the rate limiting steps and you better make some changes on these reactions or on these genes. So, I basically mentioned 2 different approaches. One based on your expert knowledge or what happened in the lab. 2, it's not based on expert knowledge, but it's based on systems biology based approach. So, which approach is better? I don't know. I'll obviously be biased. I might tell you the second approach because that is efficient and it doesn't take that much of time, but my colleagues from experimental biology they might disagree. So, does that answer the question? So, yes, the model driven approach can definitely point to the rate limiting step, although it may or may not be true, because at the end we are operating on the reaction domain. The next question that I got about the nitrogen fixing cyanose. So, the question was what is the source for carbon? I think by now we all know that, right? The source of carbon is carbon dioxide because it can photosynthesize, at least on that mode. Some of these bugs, as I mentioned, they could survive on like regular carbon sources like glucose, C5, C6 sugar. So, that is a different story, but in typical scenario if you have light, if you have carbon dioxide they can photosynthesize. So, that is their carbon source. I am really glad that some of you would like to know about opt force. Again, I could not discuss about it because of the time constraint, but what I can do and I have to discuss with you. So, if we have time after today's tutorial and if this room is available, whoever is interested to know more about all these things I can discuss. I know that some of you are interested about microbiome modeling and other stuff, so I can talk about that. So, I will talk about this if you are interested. I can let you know what are the maths involved, what are the steps that you need to get done. But one thing I can say that opt force will not be as simple as flux balance analysis. It involves a bi-level optimization. So, it basically has 2 different objectives, but yeah, we can definitely talk about it. The other question that I got is how can I ensure the stability of a pathway? Let us say you designed a pathway, how can you ensure the stability of the pathway, and how can I ensure the yield of the product? Can anyone take that? So, let us say you design a microbe and or you design like 2-3 steps in a pathway. So, you have a synthetic pathway, you have a synthetic microbe. For that let's assume that you knocked in 3 different genes. How can you make sure that your strain is stable and you are getting the yield that you initially reported down the line? How can you do that? What are the steps that you need to make sure that these things like happen, not like once, but whenever you culture that? Can you share, like if you have done it in the lab? You may not have like designed the entire pathway, but you might have designed a microbe, a gene maybe. Any answer?
Subculturing it for several generation and measuring the amount of the product in the first generation and the generation and culturing it.
Yeah. So, subculturing, yes. By subculturing you can see if if you could do that. But what I am asking, what are the steps so that you can ensure that yes, you can do it? What you are saying, it's more like detection. So, okay, I reported something, I will subculture, yes, selection pressure. If you have 3 genes incorporated with 3 different plasmids with 3 different resistance cassette, you need to maintain the selection pressure. These are the things you need to maintain, and this is very typical in this area, molecular biology, although with the So, I have two questions, so I will just finish. So, do you mind if I go a little bit over 12, like 15-20 minutes? Is it fine?
Up to 12:30.
Okay, so I'll just ask one question. Removal of contaminants from a large scale production of cyanos, I think we have discussed about it. There is no direct way of doing it, unfortunately. Energy investment in a process, again, that will depend on the techno-economic analysis. From the lab scale, you scale up, and then you have to determine what's the energy investment, because at the end you don't want to spend more energy to create some energy. It doesn't make any sense. Efficiency in the model, I think that's the last I will talk. I have few other questions, but maybe I'll talk later. How do you define if your model is efficient or not? Or in other words, how do you define if the model that you built will serve the purpose? How do you define that? Or how do you make sure that happens? Is there a linear path of doing that? Can we just build the model, test some hypothesis, build the model, generate some hypothesis, and that's it? Wet lab test. And once you do the wet lab test, what will happen? Some of the predictions will be right, some of them will not be right. You have to go back and refine the model. It's always a closed loop approach. No one could just build the model and say, well, that's it, this is the best model possible. It doesn't work like that. To make sure you are dealing with a good model, I would not say like efficient. Efficient is probably not the right word. You need to follow a closed loop approach. All right, I'll stop, and I know that I have few other questions, but probably I'm running late, so we will talk about them if I have time. So, the first lecture of today is basically to give you an overview of synthetic biology. I know that many of you are working in this area, and it's definitely an area that is I think really hot at the moment, and there is a reason why it's so hot. So, first of all, what I would like to say is, what is the difference between metabolic engineering and synthetic biology? What is the difference? Because this term synthetic biology, it was coined in 2005-2006. But you might know, or you might have seen, that some of the things that synthetic biology tries to do, people have been doing at least some of them for so many years. Then why this is synthetic biology?
All metabolic engineering thing is a synthetic biology work, or synthetic biology are not metabolic engineering, like cell signaling and other things which are associated with synthetic biology only, not metabolic engineering.
So, if I understand your answer, you are saying that there are overlaps between metabolic engineering and synthetic biology, but there are also some unique features. Is that what you are saying? Yes, partially true, but this is how metabolic engineering and synthetic biology was defined by Greg Stephanopoulos in an article published in 2012. So, basically what he is telling us that metabolic engineering is about engineering cell factories for biological manufacturing of chemical and pharmaceutical products, whereas the main focus of synthetic biology, again main focus, is fundamental biological research facilitated by the use of synthetic DNA and genetic circuits. So, I would like to bring your attention to to the second word of these two terms, engineering and biology. So, one is more application, the other one is more sciencey in that sense. So, if you look at this diagram, I am not sure if it is visible from the back. If you look at this diagram, let us say we would like to build a platform to produce a specific product, and this is a common theme that I have been discussing for the past 2-3 days, right. We are interested about building a platform, we are interested about building a platform so that we can produce a certain product. For that thing, you might have to design a pathway, or you might have to design a gene so that the pathway is complete. So, that step is known as pathway engineering. Now, to synthesize that specific protein, you might have to do some protein engineering, and then to have that protein you might have to knock in that gene, right. If I go up the central dogma of biology. So, what Greg Stephanopoulos mentioned, that synthetic biology is one of the aspects of metabolic engineering. If you look from this point of view, again the signals transduction and what else you have mentioned, that is absolutely right. But in the context of metabolic engineering, synthetic biology is one of the three major areas, like pathway engineering, protein engineering and synthetic biology. So, these again, this definition may not be the universal definition, but in the context of pathway engineering, when you are building a platform to produce a specific product, this is how these two terms are defined. All right. So, now I do not think for this audience I have to explain a lot why synthetic biology is important, right. Synthetic biology could be used for many different applications, as we have just heard about cancer drugs and what not. So, you will see its application in the biotech and biomedical areas, so many different areas. Maybe as we learned, like cancer drugs could come from a plant, a tree, but you cannot just wipe out the entire Amazon. You have to take that specific functionality, you have to borrow that, and if you can create that specific chemical or compound on a platform biosystem, let us say yeast or any other microbes, then you better do it, because that is possible. So, that is synthetic biology, borrowing something from a different biosystem and design a biosystem or a platform that we already know about. And these are some of the success stories, and I will not probably read everything out, but you could see that there are ways that more and more we could see the successful application of synthetic biology, although we would love to see more such applications. Can anyone see why sometimes we get some pushback on the application of synthetic biology? What are the issues of synthetic biology that you hear about, or you either hear from the mass or sometimes you hear from the politicians? Like, what are the issues? Ethical, what else? Horizontal gene transfer, biosafety, anything else? Loss of biodiversities. All of them are true actually. Loss of the wild type, yes. So, one of the other questions that in addition to all the issues we always get is about the control, like how confident we are that by making these changes we can confine this system in that area. So, I work in Nebraska, which is an agricultural state. Nebraska is I think the #1 corn producer in the country. The second is Iowa, the neighboring state. However, most of the corns that produce in Nebraska are not wild type anymore, because Nebraska, at least from the mid to the western Nebraska, it's semi-arid environment. It doesn't rain a lot. So, obviously, they use some sort of corn type which can survive with less water. It makes sense, because then the farmers they can grow because it's their land and they would like to grow corn. The interesting thing is they are okay with that. But if you, let's say if you are trying to sell an agricultural product or a fruits on the grocery chain, and if you don't say that it's a GMO, people will not love that. So there are issues, and I understand where they are coming from, but I think as a scientist you have to explain what are the things that we still could do. The potential is huge, and given the problems that we face, there is it would not be prudent to not to do or apply this kind of techniques to solve a problem that we could. All right. So how do synthetic biologists work? I'm sure this is something so many of you have already done. So you have a platform, you would like to engineer it. So what's the vehicle of transferring an exogenous part into that platform? What's the typical way? Plasmid. Cloning. Design a vector, design a plasmid, and make sure that the part is inside. Either you could use a replicative plasmid which could replicate, or you could make sure that that part is incorporated or integrated in the genome, as I have mentioned. If you remember, one of the examples that we have discussed is how something could get incorporated in the neutral sites. And if you remember, I have discussed that if it's incorporated in the neutral sites, then you don't need to make you don't need to have that selection pressure anymore. If you can maintain that culture, you are fine. You don't have to, like, one of the issues will be out of the picture, right? But for many organisms, and now coming back to that issue, like let's say you isolated a strain. For that kind of a system, you have no idea about the neutral system, neutral sites. So if you don't know about the neutral sites, then you cannot do these things. In addition to that, you don't know what kind of cloning is possible. For some of them, you cannot really integrate a foreign part into the genome. For some of them, so what I'm basically saying, not all the systems are naturally transformable. So you need to see what kind of system you are working on. So coming back to your question, that might be one of the deciding factor as well. I would like to do something in a given platform. How easily I can do that? Don't you think that could be one of the criteria? That could be, right?
This gene, can we integrate the gene in any neutral site, or like is there any particular choice of neutral sites?
Like one neutral site could be better than the other. So how to determine that? Again, that's an open question. So first of all, people characterize the neutral sites. Then they have to do, like they have to transfer maybe the same gene in multiple different sites and see like which one works better. So for instance, for Synechocystis 6803, 25 neutral sites have been identified, but it's only I think the 6 out of 25 we could definitively say that most of the time your transformation will be successful, but the rest, for some reason, we have no idea. They are neutral sites, but people usually have issues. So yeah, to answer your questions, no, not all of them are the same. Are you saying something?
Yeah, we have to check expression level. And
Yes. Yeah. Okay. Now we know these things, right? You can make DNA, and the cells can do the rest, right? And then synthesis of the DNA is drastically falling. I don't know how expensive it is in India, but in the US it's really, really cheap. Now we reach to a point where some of the labs, they don't design their own plasmid. They don't do anything. So basically the problem that they define is, maybe I would like to get this gene in my system. So I order a plasmid in a company, and they will design the plasmid, they will characterize, they will sequence, they will do everything, and they will send it to me. So I could quickly save some time. The caveat is I have to spend money. But then again, it's becoming cheaper and cheaper, and we all know that, like how expensive it is in India to do this thing.
Most of the time go to US. When
So if you,
synthesize a gene,
yeah,
it will go to US and expense will be almost double.
And whenever, as we have discussed, if you design an oligo, even that
That might also go if it is HPLC purified or a long oligo.
okay,
Then also it will go to US.
right. So I'm sure that down the line this will be a different scenario. I'm sure that's where we are heading to.
I just want to add, so
Yeah.
like around 2007 or 8, when I first actually synthesized my gene, and that all it was cost $2,000. Today in the same price from India, okay, we do like 5 or 6 of them.
And one of the problem is that if you buy oligo from India, which are India made,
Yeah.
that will have a problem. So you cannot be very sure that.
No, but
Because the
even
oligos which are very big, like 60 nucleotides.
Yeah. But I mean, yeah, in the US, for the oligos of that length would cost like 4 or 5 times higher than an oligo of 15 to 25. So
Yeah, same here.
that's true.
But at the end, you should be confident that it should work.
Yeah, no, that's
That
true.
is-
But I mean, what I'm saying basically, this situation will change everywhere, and I'm sure it will change here as well. So if more people do this kind of stuff in this country,
Not many people.
so if the demand is high, you better do a good job, because if you cannot get to the market, then I'm spending money to get from the US. If you can provide me good service with like lower cost, then why not, right? But anyway, so this slide I always like, because this is something that I would always like to pitch, that synthetic biologists will always have to have an engineering mind. So biologists, yes, like they do things for the sake of science, this is all good, but if you are dealing with a practical problem, you should always have to have an engineer's mind. So you need to always remember this closed loop cycle. You have to design, you have to build, you have to test, and based on that you might have to modify your design. This is an engineering principle. Usually that's how we think. So when I think about synthetic biologist, it's basically a combination of a bioengineering person with an engineering person. So if you combine biochemistry and engineering, that's what synthetic biology deserves. Now these are very crude example. Let's say you want to keep a plant hydrated, right? This can happen if you cannot do that. So what would be the engineering solution? For a mechanical engineering person, he might think about all these different issues, water and weight, and he or she might use like the tools that are out there. For an electrical engineer, it could be a different issue, like what's the conductivity, what's the light intensity, and whatnot, and the tools are there. Now for a synthetic biologist, it's a different question, right? So let's say, as I said, so how can I make sure that the plant doesn't die? So you might want to think, okay, we need to see what's out there, what are the other biological systems we can hack and put something in this plant so that this doesn't happen. So what I'm basically telling you, like, we all are trying to solve the same problem, but with different perspectives. Which one is better? That depends on the problem. It might be very case specific. So I know that many of you are working in this area. So for the next 1 or 2 minutes, I would like you to form a group of 2 or 3 and talk about what problems that an engineered microbe could solve. And you are independent to pick either a photosynthetic or a non-photosynthetic microbe. But tell me, and again, this is just an exercise. I would like to know about your thought process. So go ahead. A group of 3 or 2, you can just basically talk among yourselves in the same bench, talk about, and when I ask, you will answer me as a group. Talk. write that down, because that will be useful for the tutorial that we will do in the afternoon. So what I'll do, I will ask someone from the back, what was the problem that you are thinking about that a microbe can solve? What? Yeah. One of you can just tell me. Yeah.
So basically, talking about why we at all engineer a microbe.
Mhm.
So two of the things, one is expression and one is suppression.
Right.
Suppressing something. So basically, one is maybe we want to produce some desired product,
Mhm.
and another part is we want to curb down something which is produced by some organism.
Mhm.
Like in that sense, in a very indirect way, we can give an example of, suppose BT cotton, very
Yeah.
basic example, which actually produces after engineering BT
Mhm.
cotton. It produces the toxin,
Mhm.
which kills the bollworm.
Right.
Our ultimate aim is production of the cotton which we need,
Right.
but it's in an indirect way,
Mhm.
which kills the pest.
Yeah.
So one part is desired product from the microbe itself.
Yeah.
Another part is desired product which is growing in some other organism.
Right.
We are using the microbe so as to.
Good. Yep. Interesting problem. So you might want to write that down. Everyone should do that actually. That will be useful. What problem you thought about? This group.
Can be used as a microbes can be used for as an indicator for pollution.
As an indicator for pollution. Very good.
So it can be used for producing biofuels.
Yeah.
Then pharmaceutical.
Right. But I would say pick one problem. It could be any problem. Use.
But by engineering, we can suppress the contamination level.
Mhm.
Sir, we can also get rid of the pathogenic strain. I mean,
Yeah.
some organism, it's a pathogenic strain, but it's producing some desirable products. So
Right.
we can get that gene from there and transfer to any good strain, so that we will get the desired product, but the pathogen is not there.
Okay. Okay. So that's a good problem. Down the line, you might want to think about what are the issues that you have to encounter. Hold on to that thought. I might not talk to all of you, so I'll randomly pick few folks, and the rest of you, please write your problem down, because that will be really useful for the tutorial. What about you, folks? Yeah.
It has been seen that for
Sit down. Sit down. You don't have to stand.
engineered microbes, if we change the place, it means environmental change, then production can also be changed. And in long terms, we are dependent on the
Yeah.
single organism for the production of particular chemicals.
Right.
So if there is some environmental changes,
Yeah.
then there is a chance of less production.
Right.
Even
Mhm.
it may not work. So
Good.
it means engineered microbes should work anywhere. Then it will be more.
Right. Okay. Good. What about you folks? What have you thought about?
Me?
Yep.
Sir, various life-saving drugs were earlier very costly.
Mhm.
Using and incorporating the genes in the microorganisms are saving the cost, like insulin.
Got it. All right. So as I said, I may not be able to talk to all of you because of the time constraint, but I need you to write that down, what you have thought about, because that will be useful for the tutorial. But I'm really glad that some of you have really thought about some interesting ideas. All right. So again, you have a problem, right, which you would like to solve. So what are the tools that you have? I'm sure that many of you have already done it, right? You would like to use whatever synthetic biology tools you have. At the end, most of the times you have to make some recombinant DNA by stitching together pieces of DNA from different organisms, right? That's, if I think about synthetic biologists, that's the probably the most typical things that we do, right? So once you do that, let's say hypothetically, you design a plasmid. In the plasmid, you have a foreign gene under the control of a promoter, and now you clone that in your system. What's the next step? Expression. But also you need to also make sure that it's there the way you design without any mutations, right? How can you do that? You have to sequence that, right? So, and then we all know about PCR and what it is, right? Who doesn't know about PCR? Okay. What is PCR and why we need to do that? Why amplification is even needed?
Because quantity of DNA is very less.
Yeah.
So for experimentation.
Right. So you got your answer. We know that. How many of you have accessed this database? Registry of Standard Biological Parts. How many of you heard about iGEM, the competition? Right? So then you should have heard about this. Has anyone participated in iGEM? No. So that's the website basically we will use for today's tutorial. What it basically does, exactly like as the name suggests, it's a registry of biological parts. You will get a catalog for the promoters, for the terminators, for all other things. They have, I think, 6 or 7 different elements that they have registry for. And if you ask, they can send it to you. Basically, you have to pay, but you can get them. So these are the parts that people have used in different hosts, and most of them are well characterized and they are used in many different hosts. The good thing is you can get a list, and based on that you can decide which one is a better fit for your problem. So think it as a like you have heard me saying the database KEGG or BioPsych, if you remember yesterday. So this is kind of that kind of a database for synthetic biologists, where you can get all these different parts that are out there. Does that mean, so having this registry, does that mean that you are good for any of your problem is solved? So we will just pick parts from there and that's it, we can build something. Does that mean that? No. Many times it will happen that you will need something that is not there. Maybe you are working with a unique problem. Maybe you are working with a unique system for which you will not find anything. For instance, a newly sequenced microbe or a newly isolated microbe, you will encounter this kind of problem. But let's say for a cyano, if you work with, especially with 6803, so many things that you can get from there. Does it make sense? That's why characterized, uncharacterized, that system makes difference. What else? All right. So we all understand the engineering process, right? We have to engineer something. The first step is identifying the problem. What is forward engineering and what is backward engineering or reverse engineering? What is forward engineering? We have a problem, we will find a solution. We will test if we have found the solution, and based on that we might have to make some changes. And what is reverse engineering? We have a solution. Sorry? Yeah. Go ahead. You have a solution? Yeah. No, I mean, not in that sense, but you have an observation. What you need to know, how that happened. Bottom-up approach. So have we really discussed any reverse engineering in the class so far?
You have all the databases and build the model.
Exactly. That is reverse engineering, because you already know the phenotype. It's your job to build the framework so that you can predict the phenotype, for instance. That was reverse engineering. But for synthetic biology, we will restrict ourselves for forward engineering, because as I said, you have just few minutes back you have thought about some problems. Now it's your job to figure out a way to solve the problem. That is forward engineering. Are we clear about forwards, reverse engineering? Okay. what are the pros and cons of starting in each of the different steps in engineering process? Think about it. What are the pros or cons for design, build, test, these three different steps? What do you think, like in the design step, if you start from there, what are the positives, and if you can think about what could be the negative?
The negative could be the availability of the data that I am working with the organism. The
Yeah.
availability of the data with the organism,
Right,
which helps me to make the design.
right. But if you have, then starting from there, that could be a pro. Can anyone answer if we start from build? Is there any positive thing there? Is there any negative thing there? So what I am basically asking, if you are confused, you remember that cycle, right? You will design something, you build something, you test something. What I am asking, let's say you have started from build, and we have talked about what are the issues. Sorry, you have started from design. If someone starts from build, what kind of issue, what kind of positive or negative issues that person might face? Without, there will be lots of issues, and I sense mostly negative issues, right? Is there anything positive?
If the design is available, it is easier to make to build, because I don't need, spend time for design everything.
Or in other words, if people have designed something very similar, what you are trying to do, people have already done something very similar, so you could guess that maybe in your system that will be the same and it will be successful. That makes sense, but most of the times that's the issue. What about test? Can we start from testing something? We can. Let's say someone has built something, you can test, and again it probably stems in how well characterized the system is. You test something, you get your desired product, that's fine, but if it's it's not well characterized, someone else has designed it, then it could go like anywhere. So what I am basically trying to get here is that you really need to understand these three different steps, and you really need to understand what are the issues if you start from any of these three steps. So there is no one solution fits for all, but for most of the time it's okay if you start from the design phase, because you can always come back and modify the design. For many problems you might have to circle through this design, build, test steps few times to optimize. So that's why it's called optimize, right? So I think this is, I mean, we don't have to really discuss about it. So we all know forward engineering microbes serve a specific purpose. Why else we would like to forward engineer something? Because we already have identified a problem we would like to solve, and that's why we would like to forward engineer. One thing now I'm bringing back. So far in the past 2 weeks, past 2 days, you heard me saying about modeling. How good is model? Model is good. The way a model could help you, model could give you a list of genes that you would like to tweak. Again, model could do many other things, but I'm limiting myself to find out a list of genes that I have to tweak. So in which step modeling is helping you? Design, build, test. Which step do you think modeling is helping you? Design. Model will give you a bunch of designs. Not necessarily all of them will be right, but if you can design them, you build a system, you test, and you prove or disprove if the design was right or not. That's why modeling could be useful, because you can come up with a better design, or you can come up with a plausible sets of design which will make your life easier. Can we now all see the usefulness of a model driven approach? Because otherwise you have to design based on your expert knowledge or what you could gather from literature. Does it make sense? Okay. Now, what are the things we can design? I think this is something all of us, is there anyone who hasn't taken any biology class in his or her life? I don't think so. So, that website that I mentioned, you will get definitely a list of promoter, a list of ribosome binding sites, a list of different organisms open reading frame, or even if you don't get it there, there are other databases where you can basically find the open reading frames. But you can definitely get a list of terminators from that databases. So, there are bioparts, or there are registry of bioparts for all these different things that you need to know, or you need to design for doing a forward engineering. So, again, it's probably, I don't have to explain to this class that a gene sequence basically have all these different segments, which basically corresponds to these 4 different things, right. So, in the gene at the very beginning, you will have a promoter sequence, you will have the ribosome binding site, you will have open reading frame, transcriptional terminator, and then if we move to the translational domain, we know that RNA polymerase basically it starts here and then it ends at the transcriptional terminator, and then it produces mRNA. We know that. I am just refreshing. This is biology or molecular biology 101, just to refresh your memory. The reason why I am discussing all this, because in the next few slides I will talk about transcriptional and translational regulation. So, if you would like to regulate a gene, what kind of regulation you want to have? Do you want to control the gene in the gene expression level, or in the next level, in the central dogma, in the translational level? Which one is better than the other? I don't think anyone can give you an answer which fits for all different problems. It is very case specific. For the translation, it starts with a ribosome binding site and ends with a polypeptide folding at the end. So, this is how cell makes protein, and again this is a very crude and very simple definition of translational process. So, again coming back to that system, device, that kind of a concept that we use in control. There are electrical engineers probably here, so we always use this kind of symbols, right. So, this is how you define system, a device, a parts, and DNA, and these are the symbols you will see in the bioparts IGEM website as well. So, what do we use these symbols? These are abstractions of actual systems or actual things, right. That will make our life easier, because if you look at this symbol, you will immediately identify this is a bioparts. If you see this, you will know that this is a promoter that I am talking about. If you look this symbol, basically I am talking about a gene, right. So, that is why we use abstraction. How many engineers we have in the room? A few. Give me an example, like in your related area. I don't know what is your background, but talk to me about abstraction that you use in your field of engineering. What other abstraction we use? By abstraction I mean what other symbols that we use to represent a specific thing or a situation. We do use abstraction, right? There is a reason why we do that. Why we do that?
Because symbols are universal, so they can be understood by everybody. There is no language constraints.
There is no language. If you look at the symbol, you immediately know this is what it represents, right? What's your background? So, do you remember what other, like in what other areas you have used this kind of symbolic abstraction? All these gates, think about and, or, nor, NAND, right? We all have taken, I am sure, electrical engineering. So, that is probably the simplest example I can come up with at the moment. But what I am basically trying to say here, this is a universal language. Whenever people look at it, people will know what we are talking about. Are we talking about a device or a bioparts or a sequence? So, the reason why I am saying all this, because when you are a synthetic biologist, you need to know all these different things. So, when I talk about system, basically I have basically asked you to think about it few minutes back, that you would like to design a system to do what. So, I want a cell that detects arsenic and turns red when the levels are unsafe. So, when I asked you to think about a problem, I need you to think exactly at that level. Some of you are thinking broadly. Yes, broad problems are good, but for the scope of this class, let us limit ourselves in this kind of a problem, like this. Why am I saying this? Because this will define your system. By system, I mean what kind of microbe, and how you would like to design that. Then you can narrow things down. What you are thinking, some of you are good, useful problem, but not relevant for this class. For the sake of this class, let's limit ourselves to this kind of a problem. What kind of a system you would like to design? To do what? What kind of a device you need to have that? So, for my problem, I need to have an arsenic sensor, a color generator, because that is what I want, right? And then these are all the bio parts. We will talk about it later on, but if you look, this kind of gates, obviously you know this is AND, this is OR, this is NOT, this is NAND, this is NOR, and these are the logical truth table. We know that. We can work it out with all this Boolean logic. We know like what would be the output. We know that. This is plain and simple electrical engineering. We saw all these things. Why am I showing this? Because when you will work in this domain, when you will find out the parts, then you have to design the parts. What are the parts you need so that you can have an arsenic sensor, meaning that an arsenic sensor has to sense arsenic, and once it does that, it has to generate some color so that you know, yeah, arsenic is there. So, what kind of parts you need? That is in this level, and let us say you know these are the parts you need, and some of you are fortunate enough, you can go to the bioregistry and you can just pick and choose. Some of you will not be able to do that. You need to synthesize your own parts, and these days it is not that difficult, you can do that. But if you do not have this idea of forward engineering, that you would like to design a system, what is the next step, what is the next step, and finally this is probably the lowest level that you can get to. Your thought process will not be that easy. That is what I am saying. So, again I will skip that part. We all know what is a DNA sequence. I am not going to spend time here. So, this is the last slide of this lecture. So, basically just to quickly recap what we have discussed. We have discussed about the uniqueness of synthetic biology. We have discussed about how synthetic biology could be useful. I have given you some success stories. We also have discussed about the issues that we might face, and then we talked about forward engineering and why forward engineering could be useful to solve a practical problem. Then we have talked about the bioregistry and what are the tools that are out there and in what different level we have to operate. And then finally, I think I have given you a very practical example, a very simple example that we might all work out, or you might have already thought about it, which we will do in the tutorial in the afternoon. So, let us take a 5 minutes break.
Yeah.
All right. So, in the first lecture, basically I would like to, I tried my best to give you an overview about synthetic biology and the importance of synthetic biology, how synthetic biology could be useful to solve a practical problem, to design a system, so to speak. What is forward engineering? What are the issues that you will face in forward engineering? In this lecture, I will limit myself to the discussion of what has been going on for cyanobacteria. In many times, if I give you an example, you will see time and time again I am talking about Synecocystis 6803, and that is another piece of evidence that this is really the E. coli of cyanos, because many works have been going on in this bug for decades actually. So, again the focus of this talk is how you can genetically modify cyanobacteria, what are the many different ways you can do that, and what had happened so far. So, let us see how far we can get. If I cannot get all the slides done in this lecture, I will probably talk a little bit in the tutorial and then we will do the tutorial. The tutorial basically it's very well connected to this lecture. All right. So, by now I think all of us will agree that cyanobacteria or photosynthetic bacteria is a good platform for producing useful products, although there are some issues. One of them could be it grows slow, or most of them grow slow. For so many of them we do not have well characterized bioparts. We might have isolated some of the strains which grow fast, but the strain is not fully characterized. But all of us would agree that hypothetically, if we could design a system, then these are very good platforms. This could be very good platforms, right? So, many times you will see that cyanos, and again I am talking about a typical cyano which in the daytime can produce glycogen or store carbon dioxide in the form of glycogen, and in the night it powers its metabolism using glycogen. The good thing or the beauty of this kind of organism is you will find many different products that could only be produced in the dark time. You will find so many other products that you can produce only in the presence of light. That poses a very interesting problem. How can you engineer something, and now you have to also think about the diurnal rhythm as well, light, no light. So, it could be challenging. Now, as I said, it is a complex process, we know that, but the good thing is at least the workhorse cyanobacterial strains, they are easy to manipulate genetically compared to plant. Like I should not say easy to manipulate compared to E. coli, that is a wrong statement, but compared to plant or algae, they are easy to manipulate. And the recent advancement in synthetic biology would help you to explore many different possibilities. And interestingly, again this is something I will focus again and again. If you build a model, if you apply a strain engineering technique, that will definitely help you to forward engineer a microbe. So, that is something very difficult for a higher order system. For instance, some of the students working with Amit, you guys work in yeast, which is not a microbe, it is a eukaryotic system, it has multiple different cellular compartments, it is more complex system, because then you have to define intracellular transporters and whatnot. We do not have that kind of issue for this bug. So, the modeling framework, the metabolic engineering, in silico engineering techniques could be easily applicable for photosynthetic bacteria. Now, one of the beauties of these organisms is you can do natural transformation. So, you do not need to do any cell preparation. However, efficiency is not sufficient for using mutagenesis to create DNA libraries. All these things I will mention about 3 different techniques. All of them come with some advantage and some disadvantage. Conjugation. How many of you have done conjugation? One. Can you explain what is conjugation and how do you do it, like in probably in 30 seconds to 1 minute?
Conjugation is a process in which two cells are closely brought together and a pili-type structure is formed between them,
Yeah.
through which the genomic DNA or the plasmid DNA from one cell is transferred to the another.
Right.
And there are different enzymes which are required. I'm forgetting the enzyme name.
That is fine, you do not have to mention that, but as long as we all understand the basic principle, like forget about the details, conjugation, what that term like mean.
Joining two cells with a pipe or something like
Right.
that, through which the plasmid DNA travels to
right. So, basically you have to pass the foreign material to your system of interest through a different system when they are close by, plain and simple. That is conjugation. The issue is here, you might have to start with 3 different culture. One, your system, one, your vehicle, and one other system where you are designing the plasmid basically, right. What about electroporation? How many of you have done electroporation? Yes. How do you do electroporation, or what is electroporation?
Use some electrophorator where
So, you shock the cell and then
electrophorate the cell wall.
right. So, it is highly efficient and you do not need to wash the cell. So, these are the advantages. Now, hold on to this thought. How many cloning technique we have just discussed? 3: natural transformation, conjugation, and electroporation. Okay, are we good with this? We all understand the pluses and minuses for all of them. Now, think about it. Which method of introducing exogenous DNA is best suited for large plasmids, and why? You have a large plasmid and you would like to clone that in your system. Which method you will use and why?
We could have generally used electroporation over like calcium chloride mediated transformation for large plasmids. So that like
What? So,
that is based on experience.
no, that is based on experience, I understand, but that you can tell me, let us say you have already done it and based on that you can tell me. What I am asking, hypothetically let us assume you are a novice person, you have not done that, you only understood the pluses or minuses of all these 3 different techniques. How would you approach that? Which of these 3 methods you figure would give you higher chance to be successful?
Natural transformation.
I heard natural transformation, I heard electrophoresis. So, and I heard conjugation. So, conjugation folks, why conjugation?
It can also, because we have heard that it can also be good for genetic genome, full genome can be transferred.
Even full genome can be transported. Okay. What about electroporation? Who has said electroporation?
But then people say that electroporation creates larger pores. So
Larger pores. Yes, true.
bigger genomes.
Yeah. Do you want to add something? Okay. Anything that you would like to add?
Even so, electroporation is best suited for cells which has a thin cell wall.
So,
Easily transfer the exogenous DNA into the
right.
cell wall.
Good. Who said natural transformation? Why? Why natural transformation? Why do you think that would be preferable?
Vector is needed for
Mm-hmm.
vector is main in that means plasmid is
Right.
transferred.
Okay. So,
In 2 method there is no plasmid.
So it depends upon competence. So that is the cell is allowing.
yeah.
Cell is allowing the foreign body
Yeah.
quite convincingly.
Yeah.
That's why I think transformation is very.
No,
Okay.
but in natural transformation cell the competence level is very low. So
Right.
you have to make the cell competence by any chemical means of calcium chloride or by electroporation.
Right.
It's just an alternative for making the cell more competent.
So, here is a statement that I will make, and probably I cannot guarantee that will be a universal statement. So, based on experience, people have reported that natural transformation would be the least efficient if you are transforming a large plasmid. Most likely you will go either for conjugation or electroporation, but as I said, sometimes electroporation will have that issue that we have just discussed. Conjugation, yes, if it works, it is really, really efficient, but the problem is sometimes it does not. So, that is the issue. Many So, 3 different techniques, they come with their own advantages, but they also do have their disadvantages.
Nowadays, transfection is also used.
Transfection is also used. I didn't bring transfection here, but yeah, you are right. So, the take home message here is, whenever you are designing a system, you cannot just rely on what other people have reported. Yes, you will rely on them, but logically think about, like, if your plasmid is too big or small, then if it's too big, what are the possibilities that you might want to try? Like, logically think and then do it. But again, I would like to stress this thing, there is no one solution fits for all. It's all case specific. It all depends on what kind of system you are dealing with. However, you can operate on a generic theme. So, usually if the plasmid is big, then possibly one of these two works better. You can definitely rationalize like that. Okay. Any question? Yeah.
What if it is the other way around? If the DNA is big and the plasmid is small? Then which method would be most suitable?
I don't know the answer. Does anyone know the answer? What if the DNA is not that big, but the plasmid is big, right? Is that
The plasmid is small, but the DNA is
okay? The reverse problem. Do you see?
Then we have to choose a vector which can incorporate a larger amount of genome factors, like in.
So, are you thinking about a high copy plasmid?
No, it's not a high copy. It's like that. Those cosmid or plasmid whose own size is above 4 kb, but it can incorporate a larger amount of gene of interest. So
I see.
we can incorporate our DNA which we want to design, which we want to send. So which is a large size. We will incorporate that gene to a smaller vector.
I see.
Can itself take a large amount of.
Got it. But yeah, that's an interesting problem. I don't know the answer at the moment. I can look into that. I'll talk about it tomorrow.
What's the question? If the insert is bigger
Yeah.
than the plasmid, insert DNA.
So the question, I think the question that I asked, that was, how about if you are dealing with a plasmid which is big, but now she asked a reverse question. How about the plasmid is small, right?
and the insert DNA is
Yeah.
No, sir, the vector is designed like that.
So-
Insert DNA is the range.
So, okay, I think I understand your question. When I say the plasmid is big, basically I talk about then the size of the entire plasmid.
not the end space.
Did you understand the question? Could you repeat the question for the class again?
Like if we have a large insert size of a DNA, we can easily clone it into a large copy number or a large size of plasmid.
Right.
But what if our available choices of plasmids are of small number, a small size? Then how are we going to clone that insert DNA into a smaller
That
plasmid?
is not possible.
Why
You have to
would the
choose a
insert
bigger
DNA would be
plasmid
that much bigger?
because that is vector insert ratio.
Yeah. Why it would be bigger, that is a question, but let's say hypothetically that this is a thought experiment. So, if we are solving that problem, what would happen? I don't know the answer, but yeah, I will look into that. All right. So, as I said, now for synechocystis, all these 3 methods are possible. So, you can naturally transform, and when you naturally transform, obviously I might have mentioned it many times that you have to target the neutral sites, because that's where you want the foreign DNA to be integrated. If you are using a replicative plasmid, then there are already some replicative plasmids that is out there with RSF 1010 replicon, which works pretty good. You might so one other thing I should have mentioned. So, in synechocystis, it has its DNA, but it has I think 5 endogenous plasmids. That's a cool feature of this organism. So, you might want to try the neutral site in the genome. You can also try the neutral site in the endogenous plasmids. So, you have many different possibilities. For some transformation, for some gene, one could be more efficient than the other. And if you ask me, no model can predict that. Like no model could definitely say like, well, these are the many different possibilities. If I transform here versus there, there is a chance of higher success. At least the model that I talked about cannot do that. There might be some biophysical model. Is there a biophysical model? Do you know about any? So, I don't know. I am not an expert in this area, but what I am asking, if we could have developed that kind of a model, that could make our life even simpler, right? So, again, when you are doing natural transformation and you are integrated, you are trying to integrate the foreign part or the gene in the DNA, it is possibly by double homogeneous recombination. If you are designing a plasmid, it could be inside a replicative plasmid with that kind of a replicon or a different replicon, but that is it. So, it will go there, it will be stable, and depending on your problem, it could be a high or low copy plasmid. Which one works better? We do not know. Again, this is case specific. You cannot say for every single problem we will use high copy plasmid, because it does not work like that. What is the deciding factor between high copy, low copy? How you decide? Sorry? Origin of, no, I understand, but let us say you would like to express an exogenous gene in your system and you are using a replicative plasmid. My question is, would you use a high copy plasmid or a low copy plasmid? Do you think high copy will always work? Do you think there is a chance that in some given situation you might want to use a low copy plasmid?
Low copy with selection pressure.
So we are using, so the question was, we have to express the gene.
Right.
Then we have to use a low copy, because high copy will create a lot of protein and this could go to the inclusion bodies. So low copy will produce this in a slow pace and we can have a regular production of the protein.
So, what you are saying, that is, if I am trying to express an exogenous gene into my system, the chances of success would be better if I use a low copy plasmid, because it will take,
The expression will be better.
the expression will be better. But are we not, don't you think we are excluding some of the other considerations? What are the other considerations that we need to consider? What about you? Do you have any idea? High copy versus low copy? Have you used both for a single problem? Is there anyone who has used a replicative plasmid in your project? We have used, right? I know that some of you are working in synthetic biologies. So, you have designed a plasmid which is a replicative plasmid, right? So, when you are designing the plasmid, what I am basically asking, do you decide from the first step whether that plasmid is a high copy or a low copy plasmid, or you just design it on a backbone that is out there, or maybe some of the folks in your lab, they are using it and they are successful, so you are using it. So, did you understand my question? Do you, like, can we think about what are the other factors that we need to consider to pick a plasmid? It doesn't matter. Why? Yeah, go ahead. No, go ahead.
For our interest,
Yeah.
we want to transfer the plasmid DNA into
Right.
desired in a particular gene of
Right.
specific
Yeah.
site.
Mm-hmm.
So
No, I'm not talking about the genomic integration. I'm not talking about that.
Plasmid, whatever the copy,
Yeah.
the specific which is exogenous DNA,
Mm-hmm.
that particular attached to the particular genome.
Right. Yeah.
That's why it doesn't matter.
Okay. So, I heard about it doesn't matter. I heard about low copy is better. Do you have a different idea?
I think high copy is better because if you have a high copy number, when you're increasing the expression of the protein, you'll be producing more mRNAs of that particular machine.
Right.
So the ribosomes will be biased towards producing your protein of interest.
Right.
But then what was already pointed out by her is that if you are producing more
Yeah.
protein, there's a chance that the protein might not fold properly.
Right.
So we have to like find out.
Yeah.
It depends
So
on the type of protein being.
exactly, that's the answer I was looking for. So you cannot, for a specific problem, you cannot say just I would like to use high copy or a low copy. It depends on, you know, what kind of proteins you are making and what. So again, it's very case specific. There are ways I can definitely share some papers, like for a given problem they thought about 3 different cloning mechanism for this Synechocystis organism. So one is integrating in the genome, the other one is integrating in the endogenous plasmid, the other one with replicative plasmid. And even in the replicative plasmid, people have used high versus low copy, and they have tested everything. And again, it depends on what kind of gene you are expressing, what are the proteins. Let's say hypothetically you are creating a protein, and as you said, it doesn't fall, it doesn't degrade. It's a very energy intensive process that will create unnecessary burden on your system. So why would you want to do that? Because again, whenever you are adding an extra gene, and if that gene produces a protein, and if that protein catalyzes a reaction, and if the reaction is energy intensive, you are putting so much pressure on your system. So it depends on what kind of problem you are trying to solve. All right. Again, for this class, probably I can skip this, and we have talked about these a lot of time when we discussed model building, the central dogma of biology, and how transcription and translation works. Does anyone need an explanation for this slide? Yes? No? So if that is the case, I would like to basically point out all these different reasons, and there is a reason why we are doing it. Because let's say your model has suggested few different possibilities. One is you can mutate these 2 genes. The other is you can upregulate this gene, downregulate the other one. The other one is you can downregulate these 2 genes. So we are talking about 3 different possibilities, 3 different designs, which include mutation, up or down regulation. Mutation is mutation. You need to make sure that that gene doesn't get transcribed, right? What about up or down regulation? Basically that's when things get very tricky, because then you need to overexpress a gene. Sometimes how do you overexpress a gene? You design a tunable promoter, and then you provide more inducer, and then you might expect to express the gene more, or sometimes we add another copy of that gene. Down regulation, there are other methods, but what I am basically trying to get here is there are multiple ways you can regulate gene expression. The question is whether you would like to do in the transcriptional level or in the translational level. Which one is better? Do you think we can make a generic statement that yes, for all different applications, transcriptional regulation is better than the translational regulation? Do you think there is such thing? Or you can share for any of your work, you have done something for that specific problem, one is better than the other. Has anyone done that? No. Okay, we'll discuss. Okay, so this is the slide basically where I'm discussing everything. If I get a chance, I'll quickly explain CRISPR-Cas, although I'm not sure I will have that, but there are ways to control the gene expression in the transcriptional level. There are ways you can also control gene in the, not gene expression, in the translational level, and there basically we will talk about riboswitches. I'll talk about DNA and RNA riboswitches. I will also talk about degradation tag, how you can use that. Let's say you have a protein, but if you can tag a degradation tag with that protein. That means that as soon as it's made, it can get degraded, and that could be one of the ways you can also control how much protein you have in your system. So this slide is basically showing how many different ways you can control that, because in many times you cannot make a statement that this is what that will always work versus the other ones. It's very case specific. So let's discuss as a class what has been done in many different applications. So, CRISPR-Cas, how many of you heard about it? Very hot. What is CRISPR-Cas9? Again, there are different Cas. What is CRISPR-Cas9? What does Cas9 do? What does it do? It's endonuclease, which cuts mRNAs, DNAs. I'm just checking. But anyway, go ahead.
So it was actually discovered to be a sort of adaptive immune system in bacteria.
Right.
So bacteria gets infected by a lot of virus particles.
Right.
So what this CRISPR-Cas system, what the system has been evolved in bacteria is that if a virus attacks the bacteria, the virus will insert its genetic material.
Right.
So the bacteria takes the genetic material of that virus and it incorporates a part of the genetic material in the
Genome. Yeah.
so that if that virus infects that bacteria again, the Cas9 endonuclease will take the sequence of that virus and then by RNA interference
It will.
mutate.
So, exactly. First of all, I'm not an expert of CRISPR. I will just give you a conceptual definition here. We heard about Cas9. You might also talk about guide RNA and what it does, right? What does guide RNA do?
It guides the Cas9.
It guides. So basically, it's very intuitive. It guides the Cas9 where to fit in, where exactly in the genome it wants to fit in, and it cuts the genome. Now, one thing that I will not have time to talk, that once it does that, it cuts the genome, it could do all these things. It could replace a gene, it could knock out a gene, plain and simple, and it could also introduce point mutation, because sometimes just introducing point mutation you can achieve so many different things. So, long story short, there are two Cas9 based system that has been incorporated in two different cyanos, and these are the examples. One thing that I haven't mentioned here is that in addition to these two, the fast growing strain that I mentioned, which is Staphylococcus, what's the strain number? I forgot. 2973, yes. Staphylococcus 2973 is the fastest growing cyanobacterial strain, and Himadri's lab has designed a Cas9 system for that. So, that is a success story. So, at least for some of the cyanos, if you want to edit the genome, if you want to mutate, if you want to introduce a new genome, this system is currently working. So, that's the success stories. So, that will help us in many different applications. So, why? Because you are interested about designing a system. So, first of all, now these days, because this is so okay, before I move on, how many of you know about some issues with CRISPR-Cas9 system? There are some issues, right? What is the most prevalent issue with Cas9 or any CRISPR-Cas based system, which people always criticize? Yeah, we all know that how useful it could be for many different applications, but if you could think about one issue, what that issue is? Non-specific. Can you elaborate it and speak up so that this class could hear?
So it's a 20 nucleotide sequence that and that should have a PAM.
Yeah.
The genomic sequence.
Right.
So that the Cas9 can fit there and cut it.
Yeah.
But instead of the 20 nucleotide, if some 19
Yeah.
bases match or 18 bases match, and there is by any chance a PAM
Yeah.
to it, it's still going to cut it.
So, basically off targeting. So, you try to do something, that something may not happen. So, that is one of the issues that CRISPR-Cas always face. I mean, I know that people have been working to make it more efficient, but as soon as that happens, you will see many other applications. But for the context of cyanos, we could say that at least for few different strains, CRISPR-Cas is working, which is great. That means that we could do many different genetic manipulation. In addition to CRISPR-Cas based system, CRISPR-I is also working. Who knows about CRISPR-I? Can you talk about CRISPR-I?
In CRISPR-I, the Cas9's active site is
Right.
that it does not cleave,
Right.
but it just goes and sits there so that there is no transcription.
Exactly. So it's called deactivated Cas9. So its job is to go there and sit so that RNA polymerase cannot do anything. And interestingly, CRISPR-I based system has also been applied in a different different cyanobacteria, which is called Staphylococcus 7942. That's the strain. And I know that I'm mentioning about few numbers if you want to write down. The previous number was 2973, which is the fastest growing strain, and this one is Staphylococcus 7942, which was previously known as the fastest growing strain. And Brian Fleger's lab in Wisconsin, they first applied CRISPR-I based system in a cyanobacteria. So again, just to quickly recap what we have just discussed, we started discussing transcriptional regulation, and the first topic that we are discussing, the CRISPR-Cas based system or CRISPR-I based system, and I have given you 4 different examples where these two have already been applied. All right, what other form of transcriptional regulation? And for this next few slides I'll go faster because this is something that we always do. In the when we think about, like, if we just take CRISPR-I or CRISPR-Cas based system, if I tell you the first thing that comes to your mind, if I give you the idea of transcriptional regulation, what it is, how you can regulate transcription, what is the simplest way of doing it? Sorry? Inducer. So you will basically define a promoter which could be induced, and depending on the level of the inducer, you will get different level of gene expression. So for that kind of an application, you will find many different applications in cyano. That is not a problem. As for the inducers, these include chemicals, metals, environmental signals, metabolic state, quorum sensing molecules. How many of you have heard about optogenetics? What is optogenetics? What, like, I think it's very, if you, whenever, like, you don't know something, but if you look at the definition of the term optogenetics, opto, what does that mean? Light. Genetics part may not, like, I understand, but since most of us, I think here we understand biology or molecular biology, so that means that it has to do something with light. But go ahead. Mm-hmm. Right. Yeah, that is so you are talking about the genesis of optogenetics. That's exactly it is. But in our context, I mentioned about environmental signal. I'm basically pointing there. Environmental signal, it's a photosynthetic bug. Some of the things will be light sensitive, right? So if you could generate a tunable promoter, for instance, where you can use a specific wavelength of light as an inducer, you can do that. Then, unlike other inducer which you have to provide, you can just, flash with that wavelength of light and that's it, right? So optogenetics, there are few labs in all over the world, they are working on optogenetics, but off the top of my head, I don't know how much work has been done for cyanos. But long story short, as for the transcriptional regulation, designing a tunable promoter is something that you can always do. Can anyone think about an issue with a tunable promoter? What's the most prevalent issue of a tunable promoter or a tunable system? Do you think it's always tight? Do you think it will always work? It will work if you, if you put the inducer, if you increase the concentration, it will work. But what's the issue here? Leakiness. How do you define the off state, right? So ideally, no inducer, no expression. Does that always happen? Nope. So That is the issue with an inducible promoter system. Whenever most of the cases, at least in cyanos, it was found that the offset sometimes it's not that off. So, that could be an issue for your specific application. It might be fine for a different application, but for a specific one, even if you can get the gene to be expressed a little bit, that might create some issues down the line. How many of you have heard about riboswitches? What is a riboswitch basically? DNA versus RNA riboswitch. If you look at this diagram, what can you say? What kind of a structure this is? Hairpin, right? We know that, and we know how it forms. So, if you have this kind of a structure, you all know, like RNA polymerase will go through and it will bind there, and it will you will get your genes transcribed, right? What about this structure? If forget about this term ligand, let's assume that this is a compound. If you have this kind of a compound, your hairpin structure will be closed, and then RNA polymerase cannot bind. If it cannot bind, your gene will not be transcribed, there is no protein, that is it. This is just one of the examples. In many different applications, you will see the other way around, for which case initially the gene will not be expressed. The gene will only be expressed if you have ligand in the medium. So, again, what I am basically telling you, like there are many different riboswitches. For some of them, the default state is on, and when you have the ligand, it will be off. For some of them, it's the reverse situation. And down the line, I think few slides down, I will show you an RNA riboswitch. But is it clear from this figure how riboswitch operates? Is there any question? So, whenever I tell you about this example, these are something that already been utilized in cyanos. So, people have designed and they have found that it's working fine. What about terminators? Yes, people have used terminators, but unfortunately in the field of cyanos, no publications that characterize terminators that I can find, at least few weeks back. I don't know, something has come up yet. So, why we do care about termination? So, like we are still talking about transcriptional regulation, and that is the last part of that is the end of the transcriptional regulation, right. So, if you can modify the terminators, you can also regulate your gene expression, right. Are we clear with this? We understand that why we are talking about terminations, right, terminators. So, at least in the domain of cyanobacteria, no one has characterized this. However, for other cases, other unicellular microorganisms, there are successful applications of new termination design to regulate a gene successfully. Now, I think that is the end of my transcriptional regulation. Now, we are done with the transcriptional regulation. What is the next step? Translation, but between this post transcriptional regulation. So, antisense RNA or trans-acting RNA, how many of you have heard about them? What is so what is an antisense RNA? So, instead of 5 prime to 3 prime, we are talking about the reverse strand. So, if you use the reverse strand, and let us say there is an RNA polymerase which binds to that, and that is when antisense RNA could be made. Exactly. So, you in your system, you could make antisense RNA, you can make that. So, once you have this antisense RNA, then you can have RNAs, it could bind here and then it will cut everything, and then no transcription, or there will be transcription, sorry, no translation, my bad. So, just to quickly recap, we talked about transcriptional regulation, now we are talking about post transcriptional regulation. Logically, what is the next step? Translational regulation, right? That is what it is. Ribosome binding sites, how many Have you heard about it? We have heard about it because that's the starting point of translation, right? So, we all know that in bacterial world or in eukaryotic world, that is the probably the most prevalent start codon, right? AUG, methionine, we know that, right? And what is this sequence? AGG, AGG? Right. So, when we talk about ribosome binding sites, we know that the translation initiation rate is affected by all these three factors mostly. There are others, but these are the most prevalent one. We know that. So, hypothetically, if we could design, let us say we have a gene, we want to upregulate the gene. Hypothetically, if we could design an RBS in such a way and we could replace that with the inherent one, then there is a possibility that we might be speed up the translation. More protein, more reaction flux, maybe. So, to do that, there are bunch of tools that you can use. RBS calculator, UTR designer and RBS designer. However, there is that part. None of these tools are known to perform well for a photosynthetic microbe. As long as you work with a unicellular microbe like E. coli, gram negative or gram positive bacterium, you are fine. As far as I know, there are multiple reports where people have used RBS calculator to design RBS for at least two different cyanos. One is 6803, which is the E. coli of cyanobacteria, and the other one is 7942, the previously known fastest growing strain, and it was not successful. So, RBS calculator is good or these tools are good, but the issue is they may not work that well in a cyano. Any question? All right. So, we talked about DNA riboswitches. What about RNA riboswitches? Same principle. The only difference is now we are talking about in the translational domain. Same thing, hairpin structure, depending on the ligand, depending on the design, it could be default off and then on or the other way around. So, these kind of thing has been applied for 6803. So, for both DNA and RNA riboswitches, there are applications in cyanobacteria. Okay, should I just stop now at 12:15 or should I go on? Okay, so let's do it for 5 more minutes and then we are done. Okay. So, again we are still talking about translational regulation. Let's say you have a gene which is out. I don't think you can see it from here, but I am basically talking about that gene which is called out. So, you have a gene out, but upstream of that you have another gene called in, and these two can develop a hairpin structure, and it will basically will not let ribosome to bind and translate. And this is one situation. What about the cis repressed sequence cRNA? What about that example? What's going on in here? If you have a situation like this, obviously ribosome will not bind here, right? But if you have a trans-acting RNA, then it will bind to this part, and then you have the capability, your system has the capability for the ribosome to bind and translate. So, translational, as part of the translational regulation, there are I think 1 or 2 reports which came out fairly recently that people have applied this kind of technique in 6803 as well. I know that I am going faster and maybe you may not follow everything, but it is my job to let you know like what is out there. And again, I will pass all the slides with the reference. So, if you are interested, you can go back and check the references. Okay. Now, we talked about transcriptional regulation, post-transcriptional regulation, we talked about translational regulation. So, obviously, the last one is post-translational regulation, and that would probably the one of the most simplest thing that you can design, right. So, you have a protein, the protein will have what? What is this SSA tag? Does anyone know? So, what is a degradation tag basically in a protein? So, go ahead.
Targeting a protein,
Yeah.
and so that it can be degraded, and it's like in bacteria ubiquitination is same to this. I mean, ubiquitin is a protein which targets a specific protein,
Yeah.
and then it take the protein to hexaprotein, and that will degrade the protein.
Right. So, you made a protein in your system, and then you can make sure by tagging the protein that it gets degraded. So, if you think that for a certain gene you want to like down regulate, or you do not want any product as such, this is one of the ways in the translational level you can control that. And interestingly, for cyano there are way more works that have been done. In the database, if you remember bioparts, you will get all the degradation tags that people have used and characterized. So, if you are interested about designing this kind of a system, it is your job to go there bioparts and see what are the degradation tags that are out there. So, it is your job to pick the degradation tag, make sure that you tag that with your protein of interest, then it will be degraded, at least hypothetically when you design something. So, that is something that is useful. So, we will not do it now, but this is something I think we will start in the tutorial. So, we need to design a multi-part device that regulates at different points in the flow of genetic information. By that what I mean, can we think about a control mechanism which will let us to control the transcriptional level, post-transcriptional level, translational level, and post-translational level. So, I will stop here. Let us start from here once we come back for the tutorial. All right. Any question before we leave? No? All right. Good.
You can take a group photo.
Yeah. Okay. So, let us do one thing. Like previous class, in the next 1 minute, you will write what is the most confusing thing that you encountered in this class. Just one. What is the most confusing thing? I do not want you to write too many things, but what is the most confusing thing, or what is the thing that you do not understand yet?